Good. Okay, can I start? It's okay? Yeah, I think so. Huh?
You're recording.
Okay, I will tell you something about a new water bug species in Europe, and it's not only my work. There are numerous people involved. They are listed here in the title. And if we talk about species diversity, let's take a look on these images of different taxa. And one of the most challenging questions we all time have is how many species exist? How many species can we find on our planet? And if we make some calculations, the suggested species number is about 10 million, so more or less roundabout. And described species so far, we have only 2 million. And more than 1 million of these described species are insect species. And it's quite interesting to take a look on the rate of species description per year in this context. There was a nice summary some years ago provided by the Arizona State University. And they made a nice report, and they visualized the number of species descriptions of 2009, I think it was, in this Wordle. And you see the dominating taxon were beetles, but also some other insect taxa were quite abundant, for example, wasps, and also toolbox, of course, leafhoppers, and so on and so on. And if we take a look on the sheer numbers here, for example, we see the distribution on the different numbers and the total number here. And it's quite interesting to see that we have a rate of almost 90,000 species description per year. Let's say maybe 20,000 species description per year. It depends, of course, a little bit on each year. Otherwise, there was also a nice study showing that it takes almost 21 years in average to describe a species after its discovery. In some cases, this may be a little bit faster. In some cases, this may take even longer. Well, the most undescribed, a number of undescribed species are, of course, found in the tropics. And here you see an image of the rainforest of Peru. And if you take a look there, you see this sheer diversity of animals, of insects, of course. You see this amazing beetles here, or the butterflies, or dragonflies, damselflies, and so on, or tree hoppers. It's challenging and amazing. And of course, if we take a look in our good old Europe, we cannot challenge these numbers. But if we take a look on the smaller stuff, on the small critters in Europe, for example, this gallmichs and very small mosquitoes, we found also a lot of undescribed species for the Cecidomyidae, the Chironomidae, the Phoridae, and the Sciaridae. In Germany, at least between 1,800 and 2,200 new species. And the key to this discovery is not only the morphology, because this is quite challenging for this really, really small critters, it's barcoding. And in the past, we also did some barcoding studies for the Heteroptera, and it's almost now 8 years old. We built up a barcode library for the toolbox of Germany. About 51% of the complete fauna was barcoded. We analyzed 1,742 specimens, and we had a really cool identification rate based on the barcode index number and a species relation of 91.5%. And the quite interesting aspect of DNA barcoding is that we do not focus on one specific species, as it's done if we make some population genomics or genetic studies. For example, here highlighting some really interesting ones. No, in terms of barcoding, we try to get everything, to get all taxa that are listed and described. Of course, it's a challenge, and we are not able to get really every species, but we're on a good way. And on this context, we also switched to the semi-aquatic and aquatic Heteroptera of Germany. In our first publication, we included 46 species and a rather small number of 109 specimens. But this was increased as part of a master's thesis in 2016, as you see here. And finally, we reached another level of 67 species and 700 and 12 specimens in 2018, which was published there. And when we took a look on these data, we found for the small Pleidae minutissima, a really small water bug, 2 clusters. You see here, 8 specimens distributed finally in this monophyletic lineage, and the same here. And it's quite interesting to see that we have a really high patristic distance between the barcode data. It's about 8%, and this is really high, and something we do not expect as typical intra-specific variation, which can be found for specific taxa, of course, but this was really surprising. So what about this putative new species? So it's the pygmy back swimmer Pleidae minutissima. It's part of the Nepomorpha, and it's a Pleiad. And it's the only known species in Europe. Well, maybe 2 species, we will see later. It's really small. It has a size of 2 to 3 millimeters. It's widespread throughout Europe and also in parts of Asia. It lives in heavily weedy, stagnant or slow-flowing waters among aquatic plants. It feeds on mosquito larvae and other small aquatic arthropods, for example, Cladocera and similar things. Most individuals are flightless, and they overwinter as imago in the water. They can communicate via stridulation in both sexes. This is quite cool. They rub their rostrum on a specific structure, and they can secrete of their thoracic defenses hydrogen peroxide for protection against microbial growth. And this is really also quite cool for these small critters. Okay, and I will now show you some nice images of these really small animals, and I would like to introduce our new 3D scanning device at the ZSM. It took some time to build it up. You see it here. This is a scanner. This is the technical equipment for it, and we need also some computers for doing our 3D scans. And keep in mind, we talk about an animal which is the size of 2 to 3 millimeters. And I will now show you a small image, a small movie of this really cool scanning device, which is possible, which we can do now. Ah, this is cool, huh? Yeah, I like it. Okay, oh, sorry, wrong. Okay. So let's continue. In terms of Pleidae minutissima, we had also some taxonomic confusion in the past. It was described by Leach in 1814 in Aquatis stagnaticus propylondinum vulgatissima. It means in stagnant waters close to London in the UK. But this was not the end of this taxonomic story, because then Ray discovered subspecies. It's called Pleidae minutissima sublevis in the Provence. And McGregor and Cocker decided to describe a new species called Pleidae leachi from Scotland. Pleidae or Notonecta atomaria was described by Norvath from the Kosovo. And finally, Poisson, in his impressive activity of describing species, found also a new subspecies in Algeria called Pleidae minutissima tassili. So this is a mess of taxonomic history, and it was still unclear if the species really exist or if they are just synonyms. And thanks to Ignaz Kirchner, he checked all the species, all the data available, and he found some really impressive and highly important aspects, which are outlined here and summarized in his English summary of this Russian report. The valid name for the common Palearctic species of Pleidae is Pleidae minutissima leach. Point. Has priority over Pleidae leachi. Notonecta atomaria is placed as a synonym of Micronecta minutissima. This helps us a lot because, so now we can kick off the sublevis species, we can kick off the leachy one, and also the misidentification of Pleidae atomaria. So only Pleidae minutissima tassili from Algeria remains. Well, I did not check the material yet. It will be quite interesting to take a look on it, but well, it's from Algeria, so let's wait and see what it is. Coming back to our barcode data, we have now these two clusters of Pleia minutissima, if we use the subspecies level as well. And of course, the number of specimens we analyzed was not so high. And they are also collected from our original spot here in Germany. And in this context, we increased our number of analyzed species. As you see here, now we have here 98 specimens, and now we have here 73 specimens. And quite interesting, the split still exists. The variation or the variability is a little bit decreasing, but that's not surprising, because if we have more data, the variability will go down in terms of the interspecific variability. And it's also quite interesting if you take a look on the sampled regions. Most of the cluster A are found in the western parts of Europe. We also have now data from France and from Spain, which are not included in this presentation at the moment. But all the other ones, the clade C, they are from the continental parts. Also interesting, there are some spots where both species occur in sympathy. For example, here in Lower Saxony in Germany, and North Rhine-Westphalia in Germany, and the Saarland. And we have also some spots of the red ones of the cluster C here in the Netherlands. And OKMA will also send me some data from their barcoding studies, so we will increase our number of analyzed species as well. Okay, sometimes this may be, this variability, as I pointed out, may be an effect of some really strange effects that can be found in the mitochondrial genome. In order to analyze this a little bit in detail, we sequence from 2 selected specimens of any lineage of both lineages, a complete mitochondrial genome, and they are visualized here. And if you take a look now, we see that the distances we found for the CO1 barcode fragment was more or less also found for all other protein coding genes. For example, here ATP6, and the maximum was found for ATP8. And it's also quite interesting, if you take a look on the control region, it's completely different. We have 726 base pairs for one, and almost 1,000 base pairs for the other species. I will tell now of species, because we just not rely only on the mitochondrial data, we also check the 18S sequence data, a nuclear marker, which where substitutions clearly indicate the existence of different, but in some cases, if the substitutions are only on a small number, really closely related species. And in fact, for our Pleia minutissima cluster C and A, we found 4 substitutions along almost 2,000 base pairs. This is a clear evidence for 2 closely related but distinct species. When we talk about species delimitation, it's nice if we are able to do it with molecular tools, but if we go into the field, we do not have the opportunity to make our barcoding study all the time. So, do we have some morphological characteristics which allow us to differentiate these 2 clusters? Okay, let's take a look on the characteristics that are typically used for the Pleidae. And this is a nice collection of Parapleia species that are analyzed by Jerry Cook and co-authors. And we see, if we take a look now on these Parapleia, they can be really easily identified by different patterns of pores or setae or even coloration. Can we use it for our pleias? Unfortunately not. They are almost identical. There are some structures that offer some opportunity to delineate, but it's completely variable and we cannot find any trait in this first approach. Let's take a look at the genital plates. And this is also done for plates in a typical way. For example, here we have Parapleia melanoderma or Parapleia litura, and you see clear differences of structures, of hairs, and so, and also here Parapleia frontalis. The structures are really small, this genital plates, but they're different. Not for our ones. It's not possible to use this trait. As I pointed out, they are able to stridulate. And it's a little bit tricky. We try to stimulate some caught specimens to stridulate, but we failed because in the aquarium, we did not hear anything. And maybe there are some differences, but we do not know yet. But if we take a look on the morphological structures, we were not able to see any differences yet. So therefore also a bad smiley. Maybe there's some more power in this character, but we were not able to check it more in detail. Another one, the right gonophytes of the females, which can be also used to differentiate different species. As you see here, Neoplea notana, Neoplea punctifera, they have different structures and spines, and they can be easily differentiated. And of course, in our case, we failed again. So one of the last chances we had are the left parameers of the males, which are classically used for many, many other water bugs for the identification. And here we were successful after all. This is a left paramere, one characteristic one of the males. You see it is elongated and curved in this way. And here it's from cluster A. The differences are really subtle, but they are stable. We analyzed not only one, we analyzed, I think, almost 100 specimens in this context. And we call this here the scimitar guy because it's somewhat elongated here. And this is a so-called sickle type, curved a little bit more compact. And this is really the only useful trait we know at the moment besides the barcode data to differentiate both species. Okay, we have to take this. And we should be also aware that this is not restricted on Pleidae. If you take a look on other Nepomorpha, for example, the genus Zigara, we are also able only to identify male specimens on a really valid scale because they look almost identical, Zigara striata and Zigara dorsalis, but they can be easily identified by the different parameters, as you can see here. And not only Zigara, but this is also known, for example, for Notonecta, Euteria, and Lutea species, which are more or less common in the Central European area. And you see also here the differences of the parameters. So I'm on the end, and I will make a short summary. I think it's obvious that we face a new water dwarf beside Plea minutissima in Europe and maybe even other regions we have to check. The first clues were provided by DNA barcode data, which clearly separate both species from each other. Additional data supports these results. The morphological distinction of both species can only be made perfectly with males, as it is also possible for other water bug species. And we will use a variety of methods, complete mitochondrial genome 3D scans of the different species, more for presentation, of course, because as I pointed out, the morphological structures cannot be seen only on the animal. I use for the current species description, which is still in progress, but will be finished quite soon. So also I would like to thank again Gerd Strauss for the permission of using his really cool images of water bugs from Coriza.de, and you for your interest at this beautiful 2 Bug Tuesday. That's all.